So if Liesel's saying the screen, the computer screen is completely blank at the moment, so we're going to attempt to load it via the large screen in front. Do you know where yours is at?
It was in the lower right-hand corner.
That one there? All right. Let's see if this works.
Can people hear me with this microphone set up? Sounds good. Whoa, I can hear me now.
Awesome. Please help me in welcoming Jerry Weiland.
Okay, thank you very much for allowing me to talk to you today. I'm going to give an update on fungicide sensitivity testing in Phytophthora, the cause of Phytophthora root rot. And this is going to be some additional information over what I presented at the small fruit meeting back at the end of November. All of this is a team effort. I don't act alone. Funding came from, well, here's the special thing. Funding came from the Washington Red Raspberry Commission, as well as from the U.S. Department of Agriculture. I'm a USDA ARS researcher. My job is to study soil-borne pathogens of the small fruit industry and of the ornamental nursery industry. So I'm all about finding out what are the soil pathogens that are out there affecting production and how do we control them. So funding was from these two sources. And then Chris Benedict, Lisa DeVetter, Tom Walters, and Inga Zasada were instrumental in finding field sites for me to sample, to get the Phytophthora isolates that we work with, as well as helping sampling. Red raspberry growers, we couldn't have done this without you. Thank you so much for letting me come to your fields and take your dead plants. I really, really love the fact that you let me take your dead plants. Thank you so much. And Brian Beck and Kim Graham are in my lab, and they are the ones who conducted all the fungicide sensitivity assays. So brief outline, I'm going to talk a little bit about Phytophthora rubi, what we know about its biology, and then jump right into the study, how we determined which fields to study, what our methods were, and what the results are, and then summarize everything at the end. So background, Phytophthora rubi, it's a soil-borne pathogen that's related to algae. And because it's related to algae, it loves water. So if you have anything in your field, if you have poor drainage, if you have higher clay content, anything that promotes water staying in your field, Phytophthora is going to love that. It's very, very common in the red raspberry industry. I don't think I've found a single field yet that doesn't have Phytophthora root rot at least somewhere in the field. Of course, there are very, very severely affected fields, and there are fields where it's really hard to find Phytophthora root rot, but it is a common pathogen out there. It survives for years. It produces these oospores, which are thick cell-walled spores in the soil and in root debris, and then they are released to the soil. And because they're thick-walled, that makes them a little resistant to fungicides penetrating those. They don't degrade easily in the soil. So once you've got Phytophthora root rot, it's really, really hard to, I will say it's impossible to eradicate it. And again, damage is very, very severe on wet, heavy soils. So here's a soil with high clay content, and you can see the amount of damage from Phytophthora root rot there. So after speaking with Steve Midbow, Tom Walters, Lisa DeVetter, and a number of you growers, it sounds like there are two main fungicides being used right now to control Phytophthora root rot. One is mefenoxam, that's the active ingredient in Ridamil, and the other one is phosphorus acid, or you'll sometimes see them called phosphonates. These are, this is the fungicide that's present in the active ingredient present in things like Alliet, Agrifos, Elude, Rampart. These phosphorus acid products are in a number of things. This is a FRAC category 4, and this is 33. So they are two completely different frac things, and Tobin had really great explanation of those. Most of the data up through 2014 came from Jane Stewart, who worked in Nick Grunwald's lab. They sampled for Phytophthora in 4 Washington fields, 1 in each of these counties, Whatcom, Skagit, Cowlitz, and Clark. And they found all the Phytophthora that they got from those 4 fields were 1 species, Phytophthora rubi, and they tested 4 isolates, 1 from each county, for resistance to mefenoxam. Mefenoxam is the fungicide where you're most likely to find fungicide resistance. When you look at a number of Phytophthora studies from a bunch of different crops, mefenoxam has a single mode of action. It's one where resistance develops really quite easily. You don't usually find resistance developing in phosphorus acid, so they concentrated on mefenoxam here. And what they found is all 4 isolates were sensitive to mefenoxam. So the EC50 is a measure of how sensitive or how resistant a particular isolate is to a fungicide. So the EC50 value is 0.062 micrograms per mil. That is very, very sensitive when you consider most people start thinking a Phytophthora isolate is resistant when it's in the 100 microgram per mil range. So this is way, way, way smaller than 100 micrograms. So at the end of 2014, I was approached, maybe we need to test a more representative population of Phytophthora isolates from the Washington red raspberry industry. Up to this point, only 4 isolates had been tested. That's a very, very small number. There's a lot more Phytophthora out there. And so that was my job, is to find more Phytophthora from a larger number of fields and to test those against different fungicides. And Jane Stewart only tested against mefenoxam. And I'm going to be using the word isolate quite a bit. It's basically the same thing as individuals. So we're all individuals of the human species. If we were Phytophthora rubi, we'd all be isolates of the Phytophthora rubi species. So just a little bit of jargon that I want to get out of the way. So our methods were to go to as many representative raspberry fields as we could, and then go find diseased plants, isolate Phytophthora, find out what those Phytophthora species were, and then test them for different fungicides at different rates and find out whether resistance is out there. We targeted fields or plants where we could see symptoms. One of the key symptoms of root rot that I see is chlorosis. So you'll see this plant is quite a bit yellow than the other plants around it. So this is actually a very nice, healthy row. This was in, I think, May of last year. And this one plant, I key in on sick plants really, really well. And this plant happened to have Phytophthora root rot. It was in the early stages of going down. A little bit later, you'll often see reddening, premature fall coloration. This is again in May of last year. You shouldn't be seeing red leaves on your raspberry plants that time of year. And this is the plant itself. It obviously has some major problems there. And when I went down to the roots, I saw a lot of root rot starting. Other symptoms that are indicative of root rot is leaf scorch. So this is a sign that, or a symptom that water is not going up adequately out into the leaves. And you can imagine if a lot of your roots are rotted off, it's not going to be able to take up water as well. So you're going to start seeing damage associated with drought. Essentially, the plants cannot take up enough water, so the edges of the leaves start browning and you get these half-dead plants. Another really good symptom of Phytophthora root rot is you start seeing wilting. Again, evidence that water is not being taken up by the roots adequately to support the top growth. You'll often find fruit failure as the fruit starts to ripen or starts to form in late May, early June. These fruits are not going to do anything. And it seems like most of the symptoms of Phytophthora root rot are really bad once you get towards the June time period when the fruit starts to ripen. And that's when a lot of this wilting symptoms start appearing. But you can also see wilting. Here's a primocane wilting in September. This was also Phytophthora root rot. So really symptoms can be present. You'll see them anytime leaves are out there during the growing season. The most characteristic symptom that I see associated with root rot are these lesions that are present on the roots and then the crown. I scrape underneath the bark and I look for these reddish wet lesions, reddish brown, and you can see water starting to ooze out. This is very, very characteristic in my mind for Phytophthora root rot. This is what's actually causing the damage to the roots and preventing them from uptaking water. So this is one of the key characteristics that I see. So giving you an example, we sampled in 2015 and 2016. I'm just showing a range of the kinds of fields that we went and sampled. This is actually a very, very healthy field. Lots of beautiful green plants. I thought, oh, I'm not going to find Phytophthora root rot here. Well, after, let's say, an hour of searching, I did find a couple disease pockets. So here's one area of the field where you can see the plants have died out. They're not quite as thrifty. They have a smaller canopy, fewer canes. This was a Phytophthora rubi, Phytophthora pocket. Here's another relatively healthy field with a couple plants that are exhibiting typical symptoms of Phytophthora root rot. In this case, all the damage was located at the ends of the rows. You'll often see in probably many of your fields, the plants towards the end of your rows tend to be less healthy than plants towards the center, and these happen to have Phytophthora root rot. This is an example of a diseased pocket. So what we have, I think I'm going to stand in front of this, is we've got these healthy plants at the higher end of the field. And then as you move towards the center part of the picture, you start swooping down into a lower lying area of the field. You can see a lot of those plants are starting to die out. And then as you go back up into the background in the upper left, you're going out of that pocket and into higher ground, and the plants are healthy again. So this is just showing that the water is accumulating in these pockets, and that's where you're most likely to get Phytophthora root rot. Here's another field, abundant horsetail. Horsetail loves water almost as much as Phytophthora. And so if you've got a lot of horsetail in your fields, you've got ideal conditions for Phytophthora root rot. And often I see those two go hand in hand. Where I see a field with horsetail, I almost inevitably see a lot of problems with root rot. And this is just another two fields where Phytophthora root rot and horsetail went together. A lot of death here of the canes. There's horsetail coming up. For those of you who haven't seen the strobili starting, this is the sporulating structure produced by horsetail early in the spring. And this was another field with a lot of Phytophthora root rot. Another thing that goes hand in hand with Phytophthora root rot are heavy soils. So the more clay, the clumpier, the harder it is for the water to drain out. Here's a field with fairly heavy soils. You can see how clumpy it is. And all of these plants have died out, letting a lot of weed pressure in. And so these heavy soils, this is probably the worst field that I saw. Again, a very, very heavy soil, and these plants had abundant root rot. These heavy soils are just not ideal for growing raspberry if you've got Phytophthora present. And this is just another view of a better portion of the field. And even though these plants are still alive, you're just not going to get a lot of yield off from this. So I hope you see the range of fields that we went to try to find isolates for this study. So all in all, we went to 25 different fields, and we added in, we were able to get isolates from Nick Grunwald's group. So we have 3 additional fields from Jane Stewart's research back in 2014. So we visited 25 fields. Most of them came up from here in Whatcom County because this is where most of the raspberry production is. We did get quite a few in Skagit, and then a few from these other counties. We ended up with over 153, or over 100, let's try that again. We ended up with over 150 Phytophthora isolates. We included the 4 that were used in Jane Stewart's study, and we tested against mefenoxam, the active ingredient of Ridamil, phosphorus acid, the active ingredient of Alliet, Allude, all of those. And then right now we're in the process of testing oxythiopyprolin. It's a new product, a new fungicide that is being considered for use in red raspberry. The fungicide name is Arondis Gold. It's a brand new fungicide category that hasn't been available before. We're in the process of testing that, and we're going to test one more fungicide that is not yet registered for raspberry. But we wanted, if we found resistance to mefenoxam or phosphorus acid, we wanted to be able to have another product there for you that might be registered in the future. So if we take a look at the results, we're going from less sensitive. I've organized all these sides where the less sensitive isolates are on the left, the more sensitive isolates on the right. We've tested Ridamil, mefenoxam, all the way from 0 micrograms per mil, so free to grow as much as they want on Petri plates, up to 100 micrograms per mil. That's the high level of mefenoxam. And what we see is that although there is some variability here, the vast majority, here are all 150 plus isolates, the vast majority of them are very, 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 very sensitive to mefenoxam. That is great news. What's concerning is that there is this one little patch here, and we'll blow up that in a few minutes, that are somewhat more, I don't want to say more resistant, they're somewhat less sensitive to mefenoxam than all of these others. And the reason I'm hesitant on saying resistant, I don't want to say resistant here, is because, oh, that's, I might want to plug it in. Okay, we'll make it. So the reason I don't want to say resistant is, remember, we're testing up to 100 micrograms per mil, and generally isolates that are at, that have an EC50 value, that sensitivity value around 100, are considered resistant. Note the EC50 value here at most is almost 5. So to achieve what I would consider resistance, we'd have to go way up through the ceiling, basically, to reach 100 micrograms per mil. We're way, way down here. So these are still relatively sensitive to mefenoxam, but I want to bring in a point that Tobin brought out. All of this is Petri plate assays done in the lab. We don't know how this correlates to what you're seeing in the field. And I've heard from a number of you that you're not achieving good fungicide efficacy out in the field. We don't know whether that's due to the way the fungicides are being applied, or we don't know if maybe this little bit of variability might be what's causing fungicides not to work out in the field. So the average EC50, or the average sensitivity of all these isolates, is 0.223. That's a little higher than what Jane Stewart found, but again, we're testing over 150 isolates. She only tested 4, and most of that slightly higher bit comes from these relatively few, 25 isolates. So we're gonna blow up this area here now and just look at it in a little more detail, because there's some interesting things. I think the first thing, so this is that 150 isolates going from 5, and most of them are very sensitive. One of the most interesting things that came out of this for me is that as we started going through this, not all of the isolates that we started isolating out from the plants were Phytopthora rubi. Jane Stewart found from their 4 fields, all of their Phytopthora species belong to Phytopthora rubi. We found one field that was predominantly Phytopthora megasperma. It's a different species. And we found one other field that had a mixture of Phytopthora rubi and Phytopthora megasperma. What this means is that you've got 2 different species out there. There's evidence in the literature that Phytopthora rubi is the far more aggressive pathogen, and Phytopthora megasperma is probably a little bit less, but still able to cause root rot. However, we don't have any data on how resistance differs to these 2 species based on newer cultivar varieties. What I want to also point out is that Phytopthora megasperma, that's all of these in red, so I've got a red arrow by them, It tends to be slightly less sensitive to mefenoxam than Phytopthora rubi, so it's about 4 times less sensitive than all of the Phytopthora rubi put together. Again, we're at very, very, very low levels here. So this may not be important to the field, but we are seeing, in terms of fungicide management out in the field, this is a very, very low threshold. But it is important to know there is this difference. The other thing to notice is all these isolates in blue came from one field. We're starting to see evidence that some fields have isolates that are more resistant to mefenoxam. I shouldn't have said resistant, less sensitive to mefenoxam than other fields. So there is some variability from field to field to field in how these Phytophthora isolates are responding to mefenoxam. That may have important management implications because I have heard fungicide works great at this location, not at this location. That may be part of the reason. One last point from all of this is even at the highest rate, so even when we tested all of these Phytophthora isolates at 100 micrograms per mil, you can then take that isolate off, put it on plain media with no fungicide whatsoever, and it will start regrowing. And what that's telling me is that the highest rate of mefenoxam is not killing Phytophthora. And that is standard knowledge for us, the researchers who work on this, on root rot problems. The highest rate is not killing them, and it's often comes at a complete surprise to growers. Basically, all you're doing with these fungicides is just stopping the growth of Phytophthora for a little while. Once that fungicide wears off, they're free to grow again. And that has important management considerations. Mainly what we're trying to do is apply fungicides early in the plant cycle so that those plants have time to get big enough that they can fight off infection. And so fungicides are important for those first few years. But it is important to know these highest rates are not killing. New results. We just got in. We did a subset of all the Phytophthora isolates against phosphorus acid. We tested from 0 to 25,000 micrograms per mil. What we're finding is, for the most part, many of them are below 1,000, but we had one isolate that was far less sensitive than the others, almost 10 times less sensitive. And this, I want to go back one. Whoops, I want to go back one. F148, it was an isolate of Phytopthora megasperma. It's somewhat I'm going to keep saying that. It's somewhat less sensitive to mefenoxam. And then here, that is the exact same isolate that is quite a bit less sensitive to phosphorus acid. So we have one isolate that is not responding like all of the others to phosphorus acid. That causes me some concern. Again, Phytopthora megasperma is slightly less sensitive to phosphorus acid than P. rubi. This is other good information for us to know. One thing that I found curious is in the literature, there's not a lot out there on phosphorus acid and how resistant other Phytophthora species are to this fungicide. Most of the literature shows that other Phytophthora species are in the range of 1 to 238 micrograms per mil. Phytopthora rubi is a little bit higher than that. Phytopthora megasperma a little less. I don't know how that relates to what you're going to see with field efficacy with phosphorus acid. I heard from a number of you that phosphorus acid works better for you than mefenoxam, but we just don't know how, again, these Petri plate assays relate to field efficacy of these fungicides. And that's something to keep in mind as we go forward, that there are these differences out there. And that's where I want to end things. I think the main points that I want to bring home, the 5 things that I want you to take home, are that most of the fields out there are Phytopthora rubi. There is, however, this other species that's out there that's relatively there's a lot less known about it. There's a lot fewer studies on Phytopthora megasperma. It is important to know that there's this other Phytophthora species out there. Point 2, there's no evidence for mefenoxam resistant. Out of all those 150 isolates, All of them were really very, very sensitive to mefenoxam. Third point, I don't want to say phosphorus acid resistance. I'm saying maybe, because I don't know what that 10 times less sensitive means in terms of field efficacy of phosphorus acid. It does concern me. I just want to put out there that there's something weird going on with that one isolate of phosphorus acid. We only tested the subset, but that is something that does concern me. Another point 4, is that Phytophthora megasperma is less sensitive to both of these fungicides than Phytophthora rubi. That may have management applications, but until we start doing field studies, we're just not going to know. And then the highest fungicide rates are not killing the Phytophthora. And that's just something that I find a lot of researchers know, but that growers don't. At this point, my recommendation is to continue rotating fungicides with different modes of action. Don't just focus on one fungicide chemistry if you're going to use that. And with that, I will open it up for any questions. I think people are hungry, yeah.
In the test where you isolated the plant and you found that the plant was susceptible of the Phytophthora rubi, when it was eliminated, were the plants still exhibiting symptoms of root rot decline? I mean, there's some guesswork maybe that the root rot complex that we see is not limited to Phytophthora rubi only. That we may be seeing a complex that's causing those symptoms.
So the question is, are we seeing a complex of 2 Phytophthora species causing Phytophthora rubi or root rot out there? I would say by far most of them are Phytophthora rubi. Probably most of you who are dealing with this don't have, it doesn't look like there's a complex based on what I'm seeing and the results that we looked at and what Grunwald's lab did. Most of it appears to be Phytophthora rubi coming out. There are a couple fields where I did find, there was the one field where it was all Phytophthora megasperma, and then there was another field where it was mixed. It all looks like Phytophthora rubi. Yes, Lisa? So it's sort of an addendum to that. Are there other soil-borne pathogens that could be contributing to the disease that we're seeing? Absolutely, there could be. Absolutely, there could be. However, I'm just finishing up another study where we looked at verticillium as a potential cause of symptoms like this. Verticillium wasn't associated with disease at all. There is starting to be some new evidence that maybe a Rhizoctonia species may be associated with disease. However, nobody has studied it. So I guess the safest answer is yes, there could be other soil-borne pathogens out there causing this. However, the fact that at all of these sites where I see struggling plants like this, we get Phytophthora root rot out. I think Phytophthora is your biggest problem for dying plants. I don't think Rhizoctonia, I don't think any of these other potential soil-borne pathogens are going to cause anywhere near the damage that you see in field plots or in fields with Phytophthora. There's nothing that kills raspberry like Phytophthora rubi. So I think that is your #1 problem in terms of killing your plants. Lunchtime. Oh, one last question, Tom.
Gary, do you have any recommendations for how a grower should test the field for Phytophthora if they have Phytophthora?
So the question is, do I have any recommendations for how a grower would test for Phytophthora if they wanted to? Check with your diagnostic clinics or your local extension agents, see if they have any. Test for it. Phytophthora rubi is relatively hard to test for by traditional methods, though there are ELISA tests and qPCR tests that can be used. I am not familiar with the diagnostic labs and whether they are testing specifically for that or not.
Let's give Jared a big round of applause.